All right. So, okay, so at the beginning of this, I went, okay, this is the overall thing. We have stage 1, stage 2, stage 3. And then I talked about a little bit of terminology and built you up so you'd be able to understand what the hell is going on in those stages and wouldn't try to memorize every single pathway step individually and not recognize patterns. Okay? This is where we're going to go into the pathways. These are the Hydra heads. Okay? So they all narrow down into a body. It's a great analogy because it literally is us eating. And so we have lipids we eat, carbohydrates we eat, right? Proteins we eat. Those have initial things that are different. They're then going to narrow down, and eventually they're going to produce the same things that are all going to feed into the citric acid cycle. Okay? And you'll see them start overlapping more and more and more as you go down the body of the Hydra. Okay? And so we have glycolysis. What does lysis mean? Breaking apart. So glucose is doing what? Breaking apart. Cleaving up glucose. Cutting it up. Gluconeogenesis. Neogenesis means neo, new. Genesis, to make. Okay? What part of the Bible does God make everything? Genesis. Yeah. Neo in the Matrix meant new, right? There's also probably a Neo in the Bible, but the Matrix works easier. People remember that one better. And so we're making new glucose, right? These are going to be very counter, but they will not be a perfect overlap of the pathways. Okay? Would it be a good idea to actually have perfect overlaps of the pathways? For example, if we're taking the stairs, do we want to perfectly overlap with the people coming up? No, that's kind of a bad idea, right? And so what do you need? You need checkpoints, okay? Same sort of thing as if you have a one-way road and people are supposed to go, very narrow roads in the countryside. What do they have little cutouts on? So you can pull off, right? Yeah, very similar. So glycolysis is going to be an energy conversion pathway in most organisms. This is one of the most widespread energy pathways, okay? It probably is the most widespread energy pathway. But I don't like saying absolute statements because the field doubles in papers every 42 days. It could be the same semester and I could get proven wrong. Normally it takes a few years to get proven wrong for those say blanket statements. Not this one. Okay, so glycolysis is going to be a sequence of reactions that metabolizes 1 molecule of glucose to 2 molecules of pyruvate. This is with non-committant net production of 2 molecules of ATP. Anaerobic processes, this does not require O2. The O2 only comes in when we get back down to the citric acid cycle and on. Okay? It's common to both prokaryotic and eukaryotic cells. This means it probably was developed a long time ago, right? If it's common to both. And the same steps being common to both. Gluconeogenesis is going to be the process by which metabolic products are salvaged to synthesize glucose. Remember that these molecules you're making, many of them come out in other things as well. The metabolic process is not just using food for energy, right? You're making everything else in your body as well. Everything else you don't need to eat. So right here we have glucose. This is going to go through glycolysis. This is going to make pyruvate. From pyruvate, we actually can diverge many different directions. For example, we could ferment if we were, making beer. Let's say, okay? Did you know that happens in berries naturally as well sometimes? And so berries can have an alcohol concentration and can get things drunk. So anyways, this is fermentation. What's going to happen is your pyruvate is going to be further oxidized typically to form ethanol. And again, this is to produce a little bit more energy. We can do complete oxidation where we form CO2 and H2O. And this is what you typically have seen in your biology classes because they're more concerned with aerobic organisms like us, right? because I think fermentation is important. Obviously, Tennessee does too. but we're probably more important to us. Lactate. Anybody know what this one would be? What do you have a buildup of when you exercise too hard, too fast? Lactic acid. Yeah, that's lactate. Yeah. So this is another anaerobic pathway that happens typically in aerobic organisms. Okay. So glucose is typically consumed as starch and glycogen. It must be broken down for absorption and transport. There's actually not a lot of sources just of glucose as glucose. Okay, because the majority of it's stored, obviously, right, in the form of starch and glycogen. Alpha amylase is going to be the pancreatic enzyme that digests starch and glycogen. It's going to cleave the alpha 1,4 bonds, but will not cleave the alpha 1,6 bonds. It's going to produce maltose and maltotriose. The alpha glucosidase, maltase, this is an intestinal surface enzyme. This is going to cleave the maltose, maltotriose, and the other alpha 1,4 linked oligosaccharides into your glucose molecules. Think of it as you're going to funnel down and break it down more and more each time. Okay? And eventually you're going to get this down to, well, essentially glucose. Okay? That's going to be the majority of your sugars that you're going to break it down and transform it to. The sucrase, this is going to be an intestinal surface enzyme that will degrade ingested sucrose to fructose and glucose. By the way, this is one of the reasons why like the high fructose corn syrup and glucose would also be problematic if we ever had glucose things. But that doesn't happen. This is why those are so fattening, is because your body doesn't have to do any work. We skip a bunch of steps, right? Yeah. Okay. And so monosaccharides are then going to be transported into endothelial cells lining the intestine by active transporters. The glucose is going to move passively down its concentration gradient into the bloodstream and passively into cells that will utilize it. So the glucose transporters are GLUT1, or I like to say GLUT1, but it's not technically right. I'm sorry if you picked that up from me. GLUT1, GLUT5. Yep, it's going to happen. This is going to mediate the thermodynamically downhill movement of glucose across your animal plasma membranes. It has 12 transmembrane helix structure. Each of the member of the family has a distinctive role. And this right here is your GLUT 1, 2, 3, 4, 5. You may have wondered what the hell happened. GLUT 1 to GLUT 5. There you go. There's the GLUT 1 to GLUT 5. This is GLUT 1. It's going to be all mammalian tissues. Its Km is 1 molar. Excuse me. It's a basal glucose uptake. The GLUT 2 is going to be liver and pancreatic beta cells. Again, that's your Km there. Everybody remembers your Kms from the callus Menten? Okay, fantastic. And the pancreas plays roles in the regulation of insulin. In the liver, it removes excess glucose from your blood. GLUT 3, this is going to again be all mammalian tissues. It's a basal glucose uptake. GLUT 4, the muscle and fat cells. It's going to mount in muscle plasma membrane increases with endurance training. This is actually part of what you're doing when you're doing endurance training, is you're basically training your cells to produce more GLUT4s and to keep that level higher. Okay? That's a large part of that. I know all the training you do actually has biochemical portions to it, right? Of course. But you're literally making your cells not want to be any other way because then they suffer. So it's like taking a whip to your cells and going, you have to be this way. Or pruning, same thing. So GLUT5 is the small intestine. This is going to be primarily a fructose transporter. You may have been wondering about fructose before. The 10 enzyme catalyzed reactions make up glycolysis. Stage 1 is going to trap the glucose in the cell, modifies it so it cannot be cleaved. So it can be cleaved into phosphorylated 3-carbon units. This does not generate ATP here. Does anybody know why this step is incredibly important? Okay. I'm going to use a terrible, terrible example, but it'll be memorable. Okay? If you want to eat something, what do you have to do first? Get it to stop moving. And so typically you do that by killing it. But in the case of a molecule, you can't kill it, can you? And so how do you get the damn thing to stop moving? So you can break it apart and eat it. You trap it in the cell, don't you? And what's the best way to do that? Put a really charged group on it. Okay? Step 1, trap the glucose. Right. This is also one of the committed steps. Committed steps being ones you would have to covalently modify to get out of, right? So you've committed to this. Stage 2 is going to be oxidizes 3-carbon units to pyruvate. This is going to generate 2 molecules of ATP. Yes, this is an energy generating step. So this part of this step can actually be used to power your body even in the absence of oxygen. And so this is one of those reasons. This right here is this stage 1. So we're taking glucose. It's going to involve hexokinase. Why is it called hexokinase? Glucose has how many carbons? It's a 6-carbon chain, so it's a hexose. Hexokinase works on hexoses. It's a kinase. What do kinases do? Transfer. Yep. Yeah. Phosphorylates or transfers phosphoryl groups. You guys got it. Okay. That's what we're doing. Cool, huh? You see why we built up first those understanding what the hell the different groups of them do? Yeah. Yeah. Because it makes every other step so much easier, right? Okay, so we phosphorylated our hexose, or glucose in this case. That gives us glucose 6-phosphate. Glucose 6-phosphate is going to be worked on by the enzyme phosphoglucose isomerase. By the way, why is there only one arrow? It's not double arrow. Exactly. We can't go backwards, right? We'd have to use up energy to do that. It can't escape from the cell anymore. We're stuck there now. This is one of those committed steps. We've committed to it. This step here, is this a committing step? Do we actually commit anything to it? Can we go backwards? Yeah, this step we can go backwards, right? Okay, so this is the glucose 6-phosphate. Again, phosphoglucose isomerase. We're going to isomerize our 6-membered glucose to form the fructose 6-phosphate. Everybody see how that has isomerized it? Yeah, we've just reduced that circle, right? The cyclic compound from a 6 to a 5 member. Okay. Our heterocyclic ring. The fructose 6-phosphate is then going to be worked on by the phosphofructokinase. Again, this is a kinase, so what are we doing? Phosphorylating it, right? The substrate it works on is the phosphofructose, right? So that's the substrate. That's going to again use an ATP, and again notice that is a committed step. We use ATP to do that. It is spontaneous in that direction because of that, right? Okay, and that puts on a phosphate the other direction. Notice that we started this process with a glucose. That is a how many carbon? 6. We want to get 3 carbons, and so what do we need to do to a 6-carbon ring to make it so we can chop off 3 of those carbons and 3 of those carbons? We need to make it somewhat symmetric, don't we? Okay? Is it going to be easy to break those into equal units if one side's like gathering everything on one side? No. And so that's actually what we've accomplished here. Can everybody see that? That is actually not quite symmetric, but pretty close, right? And so now we can break this in half and we can get a symmetric, something 3 and 3. Before, we couldn't have done that, could we? No, we have no nice breaking point in there. Okay? Okay, so that's going to give you the fructose 1,6 biphosphate. The phosphates are on each side. Notice at this point what we've actually done is we've transferred phosphoryl groups to these, both sides, to make them higher energy. Okay? We set up our rock on the hill. And then now we want to break something up. What are we doing? Pushing the rock over, aren't we? Wham! And so we wham it. I know. Isn't that such wonderful chemistry terminology? Okay, so fructose 1,6-biphosphate is going to get whammed. And what's going to wham it is the aldolase. What is it called aldolase again? Anybody remember ketose, aldose? Aldose, it's an aldose. Yeah. Okay, and so we're working on an aldose, breaking it in half. There we go. This, by the way, notice what we get here. This is a what group right here? That's an aldehyde, right? Aldose. Aldolase. This one right here being what? Ketose. That's a ketone there. So the one we actually want out of this is the aldose, aldehyde. Okay? And that's the one right here. That's the glyceraldehyde 3-phosphate. The other one we get is just because we couldn't rearrange it quite enough yet, right? And so we get the dihydroxyacetone phosphate. Now, dihydroxyacetone phosphate does actually show up in other reactions and can be used and input as that, but it's not as just universally used everywhere in our body as glyceraldehyde 3-phosphate. Remember the G3P that we talked about before, which is like a universal molecule? You've actually seen this molecule in 2 pathways before because it's universal. This, by the way, needs a conversion that can go back and forth between these. And so there is an enzyme that does the conversion back and forth between these. Typically what happens is the dihydroxyacetone phosphate is converted to the glyceraldehyde 3-phosphate, and then it would go on with the pathway and use that up. Okay? And that's glycolysis, right? If it's converted to this way, we have how many of the G3Ps? 2, yeah. We make one automatically, the other one gets converted by this enzyme right here, which is the triose phosphate isomerase. Why is it called triose phosphate isomerase, by the way? These both are how many carbons? 3, tri. They're sugars, ose. Phosphates. It's an isomerase rearrangement. Yeah. And notice those classes are coming in really handy now, aren't they? Yeah. Okay. And so we end up with 2 of these at stage 1. This is why this is the stage 1. After stage 1, stage 2, you end up doing it how many times? Twice. Yeah. Now, does that necessarily mean it always goes to 2? No. If you have some process that's using this up, it's not going to end up with 2 of these. In fact, you could pull these G3Ps over to this through that pathway, right? If you needed more of that. Okay, so this is actually stage 2. This is the one that's going to happen twice most of the time. And so this one's oftentimes sort of, typically these pathways end up being done like this, with stage 1 like that, and then stage 2 going like this, backwards, so that it's nicely differentiated. Because if it wasn't on 2 different parts here, people would start getting confused. It really is a nice breakup. Okay, so twice. This is going to be where we're going to take the final product here, which is that glyceraldehyde 3-phosphate. And this one's going to go through using the enzyme, which is glyceraldehyde 3-phosphate dehydrogenase. I wonder what that does. Dehydrogenates. Remember the dehydrogenation reactions back in the Gen Chem I forced on you so much. They were so common, weren't they? Yes, that's those. That is a what type of reaction? Dehydrogenation, if we're removing hydrogens, what does that mean that reaction is? Oxidation, right? Yeah, it's a redox reaction. The other hint of that is, what does it have right here? NAD. Yeah, that's a carrier of electrons, isn't it? Yeah. Okay, and so this is going to have an orthophosphate, NAD+. These are going to be used in the reaction to form NADH, and we will have this phosphate here. See that? We had the aldehyde. We replaced it with the phosphate. Okay. Again, glyceraldehyde 3-phosphate, that's the substrate you're working on. And then this is dehydrogenation because we're removing the hydrogens. How many hydrogens did we remove? Anybody knows? Yeah, we removed one of them from here. The other one actually comes along from the phosphate. You don't see that one, but remember, orthophosphate has a hydrogen. Okay. So again, this is that 1,3-biphosphoglycerate. You may remember that from other times we had talked about like ubiquitous molecules. So again, that 1,3-biphosphoglycerate right here. The phosphoglycerate kinase is going to be the enzyme that's going to act on that. And remember that kinases can work in both directions, can't they? So in this case, instead of setting this up by putting a phosphate on, what are we doing? Taking away the phosphate and gathering that energy, aren't we? Okay? And what did you remember about 1,3-biphosphoglycerate? Did we have like a table that said that there's things that are spontaneous putting phosphates onto ADP, and there are things that are spontaneous the other direction? Didn't we? And this was on what part of the table? Putting stuff on ADP to form ADP, right? Yeah. I know, we're stitching the parts together. It's tough, isn't it? Trying to gather all those in. There's a lot. There's a lot. Like 20 years from now, you're going to go, oh, that's what that meant. I mean, hopefully you'll wake up from a dream and like, I understand it now. So the 3-phosphoglycerate is what you actually produce here. That's this one right here. Notice up here we have essentially a carboxylate, don't we? So again, that's the 3-phosphoglycerate. This is going to be worked on by the enzyme of phosphoglycerate mutase. This will give you 2-phosphoglycerate. What do we notice about this number here? From 3 to 2. Yeah. And so we just moved the phosphate group along. Mutase as in mutation change, right? So this 2-phosphoglycerate, so we're setting it up. We have our 2-phosphoglycerate. This is going to be worked by enolase. This is going to give right here this phosphoenol pyruvate. And this right here is going to remove water to do it. If you notice, we're removing water across this bond here, giving us that double bond. Hey, what is that group again? Is there something special about this? With like a double bond next to a resonance group and, can move things around, and was it maybe not stable? Exactly. Yeah, this forms that resonance structure, right? And so this is a very reactive group, and that's where the enol comes from, right here, right? So this is pyruvate with an enol group in it, and it has a phospho group on it, okay? And it's very reactive, and we remember it's very reactive in water, and so the next step's going to be a committed step again, isn't it? Because this has a lot of energy in it, very reactive. It's going to go this direction, okay? We don't need to really do anything. If you remember your table also, this was one of those very high energy ones again, okay? So the phosphoenol pyruvate again, this is going to have pyruvate kinase. Notice the pyruvate kinase is kind of interesting. When you do the kinases, typically you do the substrate that would have the lack of a phosphate. So notice our kinases, the substrate being identified in the enzyme, is these ones on the bottom for these ones, because we're removing phosphates. The other way when we're putting phosphates on, right? Okay, and so again, we enolated phosphoenol pyruvate, and then we're chopping a phosphate off that's going to give us the pyruvate right here. Okay. Does anybody have any questions? How many ATPs did we generate? 1, 2 here. 4 total. Up here? Did we generate ATP? No, we used ATP. Yeah, good. Yeah, we're using up ATP here, and then we're regenerating some, and then we actually produce extras. Yep, you got it. Yeah. So this is not an efficient way to get ATP. Now, this isn't quite as bad as you may imagine, because NADH is a form of energy as well. Okay? So that NADH can be collected up later. We'll talk about that in a bit. You need the citric acid cycle to understand that, though. So we produced more than 2 ATP in energy, but yeah, in just ATP terms, right now we've only produced 2. Very inefficient for a big old sugar molecule, right? Yeah. But that's anaerobic. Okay. So this right here, again, is your first stage of glycolysis. It's going to use the energy. Again, it's going to make 2 molecules of ATP. I know I made you do that yourself instead of reading it. My goodness, Dr. Clausen, damn evil. Okay, and so again, this is that summary. 2 ATP being used, 2 ATP being made here, done twice. Okay, and so overall net gain of 2, and an NADH. Notice these red terms are the ones that energy is being made. We're putting out energy. Blue terms being the ones we're using up the energy. Okay? And then we have the overall pathway here. Does anybody have any questions on how to read those or what we just went over? Okay, so the kinases, again, those are going to be enzymes that catalyze the transfer of the phosphoryl group from ATP to an acceptor or to ADP from a phosphoryl donor. In case you ever wonder why the hell I do that, is I want to make sure I have the correct definitions for you, so you have formal definitions. Those are actually very meaningful in the long run. Hexokinase uses ATP to phosphorylate glucose to the glucose 6-phosphate, or G6P. Almost all of the intermediates in here are used in other things and have abbreviations because they end up used in other metabolisms. Remember that these are your backbone of your metabolism. Okay? All these pathways we're going to talk about right now, the hydra as I called it, those are the backbone. Everything else will spawn from there. Oh my God, it's the god Marduk. Look it up sometime. It'll make perfect sense. So this requires magnesium 2+ or manganese 2+ as a cofactor. Oh my goodness, magnesium and manganese are so important. What should you get in your diet? Magnesium or manganese? Probably both, but at least one of them. All right. So traps glucose in the cell because, again, this G6P is going to be negatively charged. It's not a substrate for glucose transporters, and so it doesn't have any way to escape the cell. You've trapped your prey, pounce on it, right? There we go. This facilitates the metabolism of glucose into 3 carbon molecules with a high phosphoryl transfer potential. That's that table I kept mentioning. I guess I never said the name phosphoryl transfer. But you all knew what I was saying, right? Okay, and so this is the, again, hexokinase reaction. It does favor this direction because the ATP gives off more energy, right? We're going down the table, down the phosphoryl transfer. And does anybody need me to go in great detail on these specific reactions, or are you okay? Okay, I'm just going to sort of go through here and make any notes. This is going to have an induced fit for the hexokinase. So your binding of your glucose is going to cause 2 lobes of your hexokinase to move towards each other, as you can see from these figures right here. This actually makes the environment around the glucose more nonpolar, and it favors that reaction. This will exclude water from your active site to further favor that reaction. Because remember that this has a lot of hydrogen bonding around it, and we need to be able to change this out without having damn water in the way and such. Okay. Also, remember heterogeneous catalysts, what we talked about in organic a little bit? You can drive reactions by having them in environments where you don't have anything else to mess with it, and you can get better alignments. Okay? And this is one of those things that's being done. Anyways, phosphoglucose isomerase is going to catalyze the isomerization of G6P to the fructose 6-phosphate. That's the F6P. If you don't remember these abbreviations perfectly, that is okay. You can just use the other parts if you're in a problem or you're trying to remember to fill in everything else. Okay? So like if you happen to remember the enzyme and one of the reactants, the substrate, you can actually figure out everything else because the enzyme tells you what's happening, right? This is going to catalyze the conversion of the aldose into your ketose. This will take several steps because the enzyme must open the G6P ring. It'll then catalyze the isomerization, and form this F6P. You may have not realized it, but our number of carbons never changed in that process, right? It was actually just the location of the oxygen that joined the ring, okay? And that's actually that difference between aldoses and ketoses, as we're talking about back a long time ago. We talked about the sugars, right? So this will prevent the reformation of your glucose 6-phosphate, ensures that 2 interconvertible 3-carbon fragments are going to result from stage 1. If we did not do this, this end product here would not be 3 carbon fragments on both sides. Okay? We'd end up with something like 2 carbons, 4 carbons sort of stuff. Okay? And then they would not be easily interconverted, right? That wouldn't be a simple isomerization to go back and forth. Okay? Right. So this is the phosphoglucose isomerase reaction. I know we presented it as one step, but this is actually what it is. It's a multi-step reaction. Okay? And they're all equilibriums with each other, so you can go back and forth through the whole thing. Though keep in mind that the ring-closed version is highly favored, right? Remember that glucose, 99-point-something% of it is the ring-closed version. What allows us to unfold this, by the way, and make that relatively easy energetically and somewhat stable? Why does this fold up like a ring like that in the first place? Hydrogen bonding, right? When it's in a ring like this, the waters can hydrate around it and form a nice shell. So if you want to unfold it, the easiest way to do that is actually to provide it a non-polar environment, right? Yeah. So that's actually, remember all the way up back here? Oh my goodness. Try not to make so many connections so we can actually get through material. Okay. All right. And so our open chain version, we can go from the aldose to the ketose. Then when we refold it, allow it to be re-exposed to the water, it'll refold. And this will allow us to form the 5-membered ring instead, leaving this outside carbon right here. Anybody have any questions there? Fantastic. Notice again, it makes it pseudo-symmetric. The phosphofructokinase, this is abbreviated PFK, by the way, and you will see this a bunch of places. The PFK, because the fructose 6-phosphate and the fructose 1,6-biphosphate, F6P, F16BP, are incredibly ubiquitous in a bunch of other reactions. Okay? So keep that in the back of your mind that you're going to see this a lot. The PFK, this is going to be an allosteric enzyme that sets the pace of your glycolysis. This is because this is one of the first places where you have ATP going to ADP, and it's directly having a ATP in that. Notice, by the way, again, you have ATP coming in here. This right here is a lower position on our table. ATP is going to push this reaction mostly to completion. Okay? All right, anybody have any questions so far? Losing people? No. Okay. So the aldose is going to cleave the F16BP into glyceraldehyde 3-phosphate, which is abbreviated GAP, GAP. Everybody starts saying GAP, everybody ends saying GAP. So, yeah, not supposed to be GAP, I don't think, but everybody does. Dihydroxyacetone phosphate, DHAP, is its unpopular little brother. Okay? They're the twins. This one is not popular, this one is. Okay, and so the DHP is, again, that other one that's produced, and you're going to interconvert back to GAP and go along the pathway. Again, this is going to complete the stage 1 of glycolysis. After we do the next interconversion, we can move on to stage 2, but technically we can already move on to stage 2 with half of our product, right? So this is readily reversible. We can go back and forth easily in this reaction. It is a pure equilibrium because, again, this is highly used as well, okay? All three of these things show up in other reactions a lot. GAP is just even more so, okay? Only GAP can be processed to pyruvate to yield ATP. Dun, dun, dun. Yeah, so the other one cannot be directly processed to pyruvate and thus can't enter the citric acid cycle, which is where the majority of our energy is actually going to get harvested, okay, because that'll be the complete oxidation. Notice we haven't actually oxidized this that much at this point, have we? Okay. The triose phosphate isomerase, abbreviated TPI. God, the biochemists love their abbreviations. This is going to catalyze the isomerization of DHAP both ways, DHAP to GAP and GAP to DHAP. It's readily reversible, but proceeds to the GAP. This is only because in glycolysis, GAP is the one that's consumed. And so we're continuously removing that side of your equilibrium arc. Okay? Anybody have any questions so far? Fantastic. So the triose phosphate isomerase, this is actually going to be an example of the common barrel motif. Can everybody see how it looks a bit like a barrel? Okay, barrel of a gun or a potato gun, whatever you want. Less violent thing. The active site is going to be buried in your center here, and so that's your active site. Notice this is your loop here, right there. And your substrate is there. Again, we have these right here, our substrates. And these are the ones that are going to be actively involved in binding to our substrate. Okay? Fantastic. So your TPI is going to catalyze the transfer of a hydrogen atom from carbon 1 to carbon 2. This is an intramolecular oxidation reduction. Yes, it is an oxidation reduction. You are actually oxidizing one of the carbons and producing the other. And so it's kind of an interesting reaction because you haven't seen that before, have you? Have you ever seen anything like that? Yay. We get to introduce something new again, don't we? Yeah. So the isomerization is going to proceed through an ene-diol intermediate. This right here is the actual reaction, and this will make you understand the ene-diol better. What do you notice about this reaction area, by the way? It actually looks a lot like the other redox ones you've looked at, doesn't it? Same sort of side chains. Yeah, same sort of look to it. And because this is a redox. Yeah, it's a redox. It just is not involving two molecules. We just have one molecule. So this is going to be your dihydroxyacetone phosphate. And what you're seeing here is we have a carboxylate here. Again, this is a weak acid, and so this hydrogen here is more acidic than it normally would be, right? Because of the presence of the carbonyl here and the alcohol here. And this right here, this is typically what's referred to as a weak base, but in this case, it's actually going to be acting as a weak acid, okay? Because a weak base is weak acid, right? To some degree. At least in organic chemistry it is. And so what's going to happen is your double bond on the oxygen here is going to take on that hydrogen. Notice that what's going to end up occurring is we're going to transfer electrons from that hydrogen there into a double bond. That's that ene, right? And then we're going to form two alcohols. This second alcohol right there was the carbonyl, and that is the diol. And what do we remember about ene diols? They're resonance stabilized, and they can shift back and forth very easily, right? They just require the what? Loss of a proton or gaining of a proton, right? Okay. And so that's what we're actually seeing right here. Is we're forming an ene diol, and now it's incredibly easy to rearrange that because we have these groups that can take and leave protons right there. Okay, and so what's going to happen next is we are going to take a proton. Of course. So again, we are going to have these electrons here are going to bond to that hydrogen. This is going to act as a base. This is going to leave the electrons on this oxygen. Those electrons on the oxygen are going to go back into the oxygen-carbon bond. This is going to form the double bond on this side. The electrons in the double bond are going to shift over, bond to the hydrogen. This will give the hydrogen on that side. And we have rearranged which side has our carbonyl, which side has an alcohol. Yay. Okay. And again, notice that that fulfills very strictly the definitions of oxidation reduction, doesn't it? Okay. When we reduce something, what do? We add hydrogen to it, right? Right there. More hydrogen bonds. When we reduce something, we lose oxygen bonds. Went from 2 oxygen bonds to 1 oxygen bond. And the opposite happened on the other side. So you have one carbon oxidizing the other carbon. Right next to it. Shitty neighbor, if you ask me. Okay, so the roles of glue 165 and His 95. Glue 165 is going to play the role of your general acid-base catalyst. The His 95 is going to assist the catalyst by donating a proton to stabilize the negative charge that will develop on the C2 carbonyl, and then doing the opposite in the other direction when you finish the reaction. Basically, you just do the exact same thing in reverse. Okay? And so this is basically your resonance stabilized source of proton over here. Okay? Resonance stabilized, very easy to add and remove protons, right? This one right here is actually the acid catalyst right there. Okay? That you normally would do these reactions with an acid catalyst. You'd use like sulfuric acid in organic chemistry, right? That's the quote-unquote sulfuric acid. Okay? This is just the resonance structure to stabilize it, because this is not sulfuric acid, right? Not even close to the acidity. Your TPI is going to accelerate the isomerization by a factor of 10 to the 10th, compared with a simple base catalyst. The K-cat over Km ratio for the isomerization of GAP is close to the diffusion controlled limit. So this catalyst makes this reaction so fast that basically as fast as they can come here, it happens. Okay? The TPI is kinetically perfect enzyme. That's what kinetically perfect is used to refer to. It's so good, we're at such a optimal kinetic efficiency that literally if you could transport the molecules faster through the water, it would happen faster. Okay? There isn't any real time loss to this. Yeah. So it's really, really impressive. That's a huge number, by the way. Yeah. Like designing a catalyst that was that good would be amazing. That'd be like, if it was important at all, it'd be Nobel level. getting something even 10 to the 5th would be noble level. 10 to the 10th is just, that's impressive. TPI is going to suppress an undesired side reaction that would also be possible. So remember that you have this enodiol intermediate, right? What do we know about enodiols, or really just enols? Very reactive, yeah. You can fall apart really easy, right? And we have this damn phosphate here, don't we? So phosphates can fall apart very easy. So we have two very reactive groups right next to each other. You may have already kind of thought about it, but yeah, the phosphate could come off very easily, and that could then give you this methyl gloaxyl, which is the double, technically it's an aldehyde and then ketone, right? Double carbonyl, okay? And that would be fairly stable, right? So this is going to suppress that because that's hidden down here out of that environment, okay? It can't really leave easily. And so we can suppress that. We can also put groups that are going to stabilize this part of the reaction, okay? If we didn't have that enzyme doing that and this reaction were actually happening, it was kinetically happening fast enough, that would be a major product you'd make, which, shit, right? Imagine losing, half of, 25% of your product in glycolysis. So you'd have to eat 25% more sugars. Yeah. Problematic, right? Yeah, you're like, yeah, Dr. Austin, you just make me 25% more sugars. Hey, maybe that would be the answer to American obesity. We just get a mutated version of that. I wonder how toxic that is. I've never looked it up. Anyways, that's the overall pathway we've just been talking about. We've gotten to this point right here. So stage 2 is where we're coming on now. So stage 2 begins. Yay! Glyceraldehyde, 3-phosphate dehydrogenase, which is abbreviated GAPDH, catalyzes the conversion of GAP into 1,3-biphosphoglycerate, 1,3-BPG. Another really, really, really important, really common intermediate in a bunch of reactions. Okay, 1,3-BPG has a high phosphoryl transfer potential, and so this is sort of the start of where we have pushed the things up the hill, and now they can start rolling down. Right? Yeah, we already got the first effect of that, which was breaking the ring, but now we're letting things roll down the hill the rest of the way. Okay. And so here's our gap, and again, we're going to take NAD plus and a free phosphate. The free phosphate is going to come on here, and we'll take the hydrogen on the NAD for the NADH. Notice that every time we talk about NAD plus and NADH, a lot of biochemists will actually call this NADH2 plus, right? And that's why. Can everybody see that okay? It almost always is accompanied by an H plus. Anyways, everybody see the phosphate would have had the other H, right? Yeah. Okay. So your process 1 is going to be the oxidation of the aldehyde to a carboxylic acid. This is by the NAD plus. This is thermodynamically favorable. It has a delta G naught prime of approximately negative 50 kilojoules per mole. And so this will actually go through to this form right here. It will leave an alcohol on there when it's happening, okay? It's another one of those one biochemistry steps that's actually like 2, 3, 4 organic chemistry steps. Yeah. The process 2 is going to be joining of the carboxylic acid and orthophosphate. This will form the acyl phosphate product, thermodynamically unfavorable because you're adding that phosphate on. And it has a delta G of approximately 50 kilojoules per mole. What do you notice about these numbers? negative 50, positive 50. Gives you an overall 0. 0 means equilibrium, doesn't it? Yeah. And that's what forms your equilibrium. If you only were doing one of those, it wouldn't be an equilibrium. So this is going to give us our acyl formation hydration. Notice we are removing water across this. Technically, the hydrogen's there. We're removing water because the OH comes off. The glyceraldehyde 3-phosphate dehydrogenase reactions are going to be coupled. The reaction is going to be linked by the formation of an energy-rich thioester intermediate in the activation site of the GAPDH. What time is it, by the way? Oh, is it time? Let's stop here. I actually need to run by my house real quick because apparently my